For this section, we're going to try to answer 3 questions, and they are, why does the stomach produce acid, and how does it know when to produce acid? How does the pancreas know when to secrete digestive enzymes and bicarbonate into the small intestine? And how does the gallbladder know when to secrete bile to help with lipids? And first, we need to know the 6 types of epithelial cells that are in the stomach. The first one is chief cells. They secrete pepsinogen, which is the inactive form of pepsin, and they secrete that into the stomach lumen. The pepsinogen is converted to pepsin when there's acid. So when something increases the acid production, it also increases the pepsinogen secretion as well. The second type of cell is a mucus-secreting cell. So obviously it secretes mucus, which is important in helping protect sensitive cells in the acidic environment of the stomach. The third cell is enterochromaffin-like cells, and these release histamine into the stomach lumen, which helps with acid production. And then the fourth cell is G cells, and these produce hormone gastrin, and this is released into the blood, and it circulates in the blood until it finds receptors found on certain cells to bind to. And G cells, they are stimulated to release gastrin in response to stomach stretch or acetylcholine from the nervous system, amino acids, and also pH changes that are greater than 3. The fifth cells in the stomach are D cells, and they're found in the stomach, intestine, and the pancreas, and they release somatostatin, which inhibits the parietal cell, G cell, and ECL cells. And also the ECL cells are the enterochromaffin-like cells, just FYI there. And then the 6th type of cell in the stomach are parietal cells, and these secrete acid and intrinsic factor into the stomach lumen. And there we go. The intrinsic factor is required for vitamin B12 absorption in the small intestine, and without this intrinsic factor, someone would become very anemic because vitamin B12 is really important in red blood cell production. The parietal cell has lots of proteins that help regulate acid production. So on the basal side of the parietal cell is a bicarbonate chloride exchanger. And then on the apical side of the parietal cell, there's a hydrogen-potassium ATPase pump and also a chloride channel. So those are the 2 sides of the cell and what they have. So when a parietal cell is stimulated, the hydrogen-potassium pump pushes out hydrogen into the stomach lumen in exchange for potassium. And then the chloride that's absorbed by the blood in exchange for bicarbonate is secreted into the lumen also, but it's done by chloride channel. And the hydrogen used for the pump is provided by the entry of carbon dioxide, and the conversion of the carbon dioxide to the bicarbonate and the hydrogen ion is done by the enzyme carbonic anhydrase. Sorry, I hope you can still see that. So the stimulation of parietal cells happens directly through actions of gastrin and histamine binding to membrane receptors on parietal cells. But acetylcholine and gastrin can regulate secretion indirectly by acting on the enterochromaffin-like cells to release histamine. So there's directly and indirectly. And they've also come up with drugs that help decrease acid production, and these are like histamine receptor blocker drugs, and also drugs that block the hydrogen-potassium ATPase activity. So that was the first question. And then the second question, which was, how does the pancreas know when to secrete digestive enzymes and bicarbonate into the small intestine? And so there's 2 types of cells. There's the S cells and the I cells that are involved with this, and these cells are in the small intestinal epithelium. And so the S cells, these release hormone secretin into the blood. Sorry, I need to turn this into the bloodstream. And it's released in response to acid from the stomach, and it acts in different ways. So the first way is that this S cell, it binds to receptors on pancreatic cells and causes release of bicarbonate, which is released into the duct system of the pancreas, and it ends up emptying into the small intestine. So then the bicarbonate that's in the small intestine helps buffer the acidic pH of the stomach. And then the S cell also interacts with G cells to decrease the secretion of gastrin, which slows the acid production. And then the third thing that the S cell does is it stimulates bile production by the liver. Okay. And the I cell, it releases the hormone cholecystokinin into the bloodstream. And its release of this cholecystokinin, is regulated by acid and lipids. And this acts in different ways also. The first way that I cells act is that they interact with the pancreatic cells to increase release of digestive enzymes. They interact with cells of the gallbladder and induce contraction of the gallbladder, which releases bile into the small intestine. And that's actually the answer for the third question that we had at the beginning. The third question asked, how does the gallbladder know when to secrete bile to help with lipids? And then that just answered it. So the I cell interacts with cells of the gallbladder and induces contracting of the gallbladder, which releases the bile into the small intestine. So that's the answer for the third one. And then the third way that I cells act are that they interact with the pyloric sphincter, causing it to contract, which decreases stomach emptying. And then the fourth way is that the I cells interact with our brain somehow to make us feel full. And then there's also K cells, which are important. And K cells, they release gastric inhibitory peptide, so GIP, and this binds to pancreatic beta cells. So K cells stimulates release of insulin in preparation for the glucose that will enter the blood from the small intestine. And that's it's just like a lot of information, but at the end of the study guide, it gave a hamburger example, and it helped me a lot, so I was just going to walk through that really quickly. So say you walk into a hamburger joint, and so the first thing that will happen is that our olfactory system will stimulate, and our brain will send the signals, which would be acetylcholine, to our digestive system to get it ready. And the certain parts that are getting ready are salivary glands and our stomach cells, which are again G cells, ECL cells, parietal, and chief cells. And when your mouth starts to salivate before you even start eating, that's called the cephalic phase of digestion. So as the stomach starts to enlarge because you're eating, and there's a decrease in acid because the food is soaking it up, and it stimulates more gastrin released by G cells and increases the release of acid by parietal cells, and also increases the release of pepsinogen by chief cells. And then the D cells will also release somatostatin as a type of brake to slow this process down. It won't stop the process of digestion, but it just kind of slows everything down. And the partial digestion of carbohydrates begins in the mouth by salivary amylase, but then as the pH in the stomach goes back to being more acidic, the amylase is inactivated. And the pepsin becomes more activated in acidic environment, so that will begin to break down the proteins in our food. And that takes about 3 to 5 hours for the hamburger to be completely cleared. So then the hamburger will move from our stomach to our small intestine, and the S cell will respond to the acid by releasing secretin, and then the I cell will release cholecystokinin, and the K cell will release the GIP. And the cholecystokinin will stimulate pyloric sphincter to contract, slowing the gastric emptying. And then it'll just take 3 to 6 hours to go through the whole small intestine. And then once it gets into the large intestine, it'll take about 30 to 60 hours to be excreted from the body.